Hello and hello everyone, welcome to class 12 biology. In our previous video we discussed about restriction endonucleases as one of the important tools of recombinant DNA technology. Now in this video we will be discussing about other two important tools of recombinant DNA technology. They are cloning vectors and competent host. So what is a cloning vector? A cloning vector is a small piece of DNA that can carry a foreign DNA fragment into a host organism. And within the host organism, it can be stably maintained for cloning purposes. I have told you before, vectors are those DNA molecules which can carry our gene of interest into a host organism, thereby transforming the host organism into a genetically modified organism. Right, I have told you about plasmids, right? And in earlier days, bacteriophages and plasmids, they were used as cloning vectors. Why? Because bacteriophage DNA and plasmid DNA, they have the ability to replicate autonomously within the bacterial cells, right? And because of that, they have got high copy numbers within the bacterial cell. What do we mean by copy number? Copy number is their number per cell, right? Bacterial DNA and the plasmid DNA, they autonomously replicate within the bacterial cell, thereby keeping their numbers high. And this is a desirable trait because if we can insert a gene of interest within these DNA and introduce it into a bacterial cell, these plasmids and these bacteriophages, they will replicate within the bacterial cell autonomously and keep the number of our desired gene high within the bacterial cell, right? So that is a desirable quality of a cloning vector. But nowadays, scientists, they use genetically engineered vectors instead of bacteriophages and plasmids. Genetically engineered vectors are those vectors which are created in the laboratory by scientists themselves to be used specifically for creating recombinant DNA. Right? Genetically engineered vectors, they are good for easier linking of foreign DNA, for easier selection of recombinants from non-recombinants, and therefore the use of genetically engineered vectors are preferred over the use of bacteriophages and plasmids. I will explain these two words later on, recombinants and non-recombinants, right? So don't worry about it right now. One such genetically engineered vector is PBR322, right? It is widely used in different laboratories world over, right? So PBR322 is the name of a genetically engineered cloning vector. So this is the diagram of PBR322. It is given in your textbook on page number 199, figure number 11.4. You should practice drawing this diagram and try to understand this particular diagram. Right, this is important from your examination point of view. So PBR322, right? It is a genetically engineered cloning vector widely used because it has got certain characteristics. So let us try to understand what are some desirable characters which is required within a cloning vector. Now there are some important features that are required in a good cloning vector. And the first important feature that is required in a cloning vector is origin of replication, or ORI in short. Origin of replication is a sequence of DNA where DNA replication starts. And it also controls the copy number of recombinant DNA within the host organism. If we take a look at the diagram of PBR322, the genetically engineered cloning vector, the blue region over here, it represents the origin of replication. It is the DNA sequence where the DNA polymerase will come and bind and initiate the replication process, thereby creating multiple copies of itself within the host organism. Right? Now, if this vector was carrying our gene of interest, a foreign gene of interest, along with the vector, the foreign gene of interest will also get cloned. So that is the desirable outcome. For that to happen, the origin of replication should be there on the cloning vector. So the first important feature is origin of replication. The second important feature that is required in a cloning vector is selectable markers. So selectable markers are genes which can help in identifying transformants from non-transformants. Now, what do we mean by transformants and non-transformants? In order to understand that, let us first try to understand transformation process. Transformation process is the introduction of foreign DNA or foreign gene into a host bacterium, thereby transforming the host bacterium into a genetically modified organism. Since we have introduced a foreign gene into the host bacterium, that will change the phenotype of the host bacterium, thereby making it into a genetically modified organism. Right? And transformants and non-transformants means transformants are those bacteria which now contains the foreign gene. Right? And non-transformants are those bacteria which does not contain the foreign gene. So that's the difference. In order to understand this one better, do you recall we saw this particular picture in our previous video, right? This particular petri dish contains two different colonies of bacteria. The one which glow under UV light, they are called as transformants, and the one which does not glow under UV light, they are called as non-transformants. Why? Because these bacterial colonies, they are glowing because they contain the foreign gene, GFP gene from jellyfish, green fluorescent protein gene from jellyfish, right? Therefore, they are able to glow under UV light. And these bacterial colonies which does not glow under UV light, they do not contain this particular gene from jellyfish, and therefore they are not glowing under UV light because they are not producing green fluorescent protein, right? So the difference between transformant and non-transformant is transformant contains the foreign gene, whereas the non-transformant, they do not contain the foreign gene. Okay, now in this case, the cloning vector itself is the foreign DNA for the host bacterium. Remember, the cloning vector does not belong to the host bacterium. Scientists, they are trying to introduce cloning vector into the host bacterium. So for the host bacterium, cloning vector is a foreign DNA. Now when the scientists, they try to introduce cloning vector into bacteria, some bacteria, they take in a cloning vector, thus becoming transformants, and some bacteria, they fail to take in cloning vector, thus remaining as non-transformants. Now how do we identify transformants from non-transformants? Now here we have antibiotic resistant genes as selectable markers, right? And selectable markers help in selecting transformants. If you look at the picture of PBR322, the two purple regions over here, they are antibiotic resistant genes. Here we have ampicillin resistant gene, and here we have tetracycline resistant gene. Ampicillin and tetracycline are two antibiotics. Okay, so these two genes, they are used as selectable markers. Remember, the original host bacterium, they are not resistant to these two antibiotics. If the host bacterium becomes transformant, if the host bacterium takes in this particular cloning vector within it, the host bacterium will develop antibiotic resistant capability, right? So these two genes, ampicillin resistant gene and tetracycline resistant gene, they can be used as selectable markers, which means these can help in selecting the transformants and identifying transformants from non-transformants. So selectable marker is the second important feature that is required in a cloning vector. The third important feature that is required in a cloning vector is cloning sites. A good cloning vector should have cloning sites. What are cloning sites? They are the recognition sites for restriction enzymes. And we all know that restriction enzymes are molecular scissors which can cut the DNA at their recognition sites, right? And we use restriction enzymes in order to create a recombinant DNA. If we take the example of PBR322, which is a cloning vector, it has got all these recognition sites over here for these restriction enzymes. We all know that EcoR1 is a restriction enzyme. The recognition site for EcoR1 is right over here. If we use EcoR1 in order to create a recombinant DNA, the EcoR1 will make a cut right over here, and we can attach the foreign DNA right over here and create a recombinant DNA, right? So that is the necessity of cloning sites. Now, the thing over here is a single recognition site for each restriction enzyme is preferred. Why? Why there is only one recognition site for EcoR1? Why there are no multiple recognition sites for EcoR1? That is because if there are multiple recognition sites for a restriction enzyme, the restriction enzyme will make multiple cuts on the vector. That will create small pieces of the vector, which is not a desirable result. What we need to do is we need to just make one single cut so that we can introduce the foreign gene and create a recombinant DNA, right? Getting a multiple pieces of cloning vector is not useful for creating a recombinant DNA. So therefore, we have got only one a recognition site for one restriction enzyme. So that is the desirable trait. Now this is important, please pay attention. The restriction sites present in the two antibiotic resistant genes can be used to insert foreign DNA. For example, if we use BAMH1, whose recognition site is located right in the middle of tetracycline resistant gene, the BAMH1 will make a cut right over here, and we can insert a foreign gene or a foreign DNA right over here. What this will do is, this will inactivate the tetracycline resistant gene, and the process is called as insertional inactivation. The term itself is self-explanatory. Due to insertion of a foreign DNA right in the middle of tetracycline resistant gene, the tetracycline resistant gene will be inactivated, and that is called as insertional inactivation. And when a host bacterium receives such kind of recombinant DNA, the host bacterium will be ampicillin resistant, but the host bacterium won't be tetracycline resistant. Why? Because the tetracycline resistant gene has been inactivated due to insertion of foreign DNA right in the middle. Right. So this is important in order to identify recombinants from non-recombinants. Earlier we talked about identification of transformants from non-transformants. Now we will talk about identification of recombinants from non-recombinants. Let us now try to understand the process of identifying recombinant bacteria from non-recombinant bacteria by going through a series of diagrams. In this particular diagram, you can see the initial stages of creating a recombinant DNA. So our main goal is to create a recombinant DNA. In order to create a recombinant DNA, we take a mixture in which it contains a plasmid or a vector like PBR322, and we take the foreign DNA containing the gene of interest. Then we treat them both with the same restriction enzyme. Let's say we use BAMH1. If we use the same restriction enzyme, it will create complementary sticky ends, thereby making it easier for us to attach this foreign DNA to the plasmid. And this will result in the formation of recombinant DNA. Now remember, since we used BAMH1, it will insert the foreign DNA right in the middle of tetracycline resistant gene, thereby inactivating the tetracycline resistant gene. Now this is called as recombinant DNA. Now in the mixture, we also have those plasmids or those vectors which have failed to take in the foreign DNA, and they are called as non-recombinant DNA. And now when we introduce the bacteria host organism like E. coli into the mixture, what can the E. coli do is, E. coli can either take in the recombinant DNA or the non-recombinant DNA, or it can take in none of the two, right? So this will result in the formation of non-transformants and transformants. So how do we identify non-transformants from transformants? Non-transformants, they won't be resistant to both ampicillin and tetracycline. Why? Because they do not have these two genes, ampicillin resistant and tetracycline resistant genes, right? So by using ampicillin and tetracycline, if the bacteria dies, we can simply reject them, right? So now we are left with these two, right? These two transformants, and one of the transformant is with the recombinant DNA and one of the transformant is with non-recombinant DNA. So how do we identify between these two? So that is the main idea, right? That is the main goal in order to understand, right? So we need to understand this, right? Now, if you know, if you recollect, we used BAMH1, right? If we use BAMH1, it will introduce the foreign DNA right in the middle of tetracycline resistant gene, right? Therefore, the recombinant DNA will have inactive tetracycline resistant gene, and therefore the bacteria will be resistant to ampicillin, but it will not be resistant to tetracycline. So if we use tetracycline, the bacteria will die. On the other hand, the transformant bacteria, which contains the non-recombinant DNA, will be resistant to both ampicillin as well as tetracycline, right? So by using this particular feature of antibiotic resistance, we can select the recombinant from non-recombinant. Okay, is it clear? Identification of recombinant and non-recombinant bacteria. So that is the main idea of using BAMH1 in order to insert this foreign gene right in the middle of tetracycline resistant gene, thereby making the tetracycline resistant gene inactive. And we are using this particular insertional inactivation property in order to identify recombinants from non-recombinants. So now you must have realized that the process of identifying recombinants from non-recombinants due to inactivation of antibiotic resistant genes is a very cumbersome process. It's a very complicated process. Even the scientists, they realized the same thing. And therefore, the scientists developed alternative selectable markers, right? So instead of using antibiotic resistant genes as selectable markers, they used different genes. And those different genes are genes which codes for enzymes such as beta-galactosidase. And these enzymes, in the presence of chromogenic substrate, they produces colored reaction. And this property of producing colored reaction in the presence of chromogenic substrate is used for identification of recombinants from non-recombinants. How did they do that? When a foreign DNA is inserted into such a gene, which codes for the enzyme, the gene becomes inactive due to insertional inactivation and does not produce the enzyme. And the bacterial colonies which does not produce any color can be identified as recombinant colonies. For example, in this particular picture, you can see in a petri dish, the growth medium contains chromogenic substrate for beta-galactosidase enzyme. And we see two different types of bacterial colonies, one which shows colored reaction, this blue color, and one which does not show any colored reaction. So the bacterial colonies which are showing the colored reaction is called as non-recombinant. They can be identified as non-recombinant colonies. And the bacterial colonies which are not showing any colored reaction, they can be called as recombinant colonies. Why? Because inside the recombinant colonies, right, the genes which codes for beta-galactosidase has been inactivated due to insertion of foreign DNA, and therefore they are recombinant colonies. And in these non-recombinant colonies, their beta-galactosidase gene is not inactivated, right? So they are non-recombinant colonies. So I hope it is clear, right, how the alternative selectable markers such as genes which codes for enzymes such as beta-galactosidase is a good option against the use of antibiotic resistant selectable markers. Okay. The fourth and the final important feature required in a cloning vector that is mentioned in your textbook is vectors for cloning genes in plants and animals. So the specificity of vectors for cloning genes in plant cells and animal cells is another important feature. Now, for thousands of years, different pathogens of plants and animals, they have been developing different tools or different methods through evolutionary process of injecting their own gene into the host organism. Scientists nowadays, they are trying to modify those tools developed by the pathogens, and they are using those tools to deliver our own gene of interest within the plant cells and animal cells. For example, in plant cells, we can introduce our gene of interest by using modified tumor-inducing plasmids of Agrobacterium tumefaciens. So Agrobacterium tumefaciens is a pathogen of different plants. They can induce tumors by injecting tumor-inducing plasmids. Scientists, they have modified tumor-inducing plasmids so that they can no longer cause tumor, but they are using that particular tool to deliver our gene of interest into the plant cells. And the same thing for animal cells as well. We have got retroviruses, which is known to cause cancer within the animals, right? So retroviruses, if we disarm the retroviruses, we can deliver our desirable genes into the animal cells as well. You know how retroviruses infect the animal cell. We have studied about HIV, right? So for plant cells, we use tumor-inducing plasmids of Agrobacterium tumefaciens, and for animal cells, we use disarmed retroviruses. So these two names are important. With that, we have finished discussion about important features required in a cloning vector. So there are 4 different features. So you have to remember them. The third important tool in recombinant DNA technology is a competent host. A competent host is a host organism which readily takes up recombinant DNA. After we create a recombinant DNA, we need to insert this recombinant DNA into a host organism. Right? And the most commonly used host organisms are bacterial cells, especially the E. coli bacteria. Now the bacterial cells, they do not readily take in recombinant DNA. Why? Because DNA being hydrophilic molecule, they cannot easily pass through the lipid bilayer of bacterial cell membranes. And over that, the bacterial cell also have got a cell wall. So therefore, the bacterial cells, they are not competent host. We should make the bacterial cells competent to take up the recombinant DNA. How do we make the bacterial cells competent host? Right. In this particular process, we first treat the bacteria with a specific concentration of a divalent cation such as calcium. This will make the bacterial cell walls porous to the recombinant DNA. After that, we incubate the cells with recombinant DNA on ice, followed by placing them briefly in 42 degrees Celsius, which is called as heat shock, and then back on ice. This will enable the bacterial cells to take up the recombinant DNA. This will make the bacterial cells competent host. In this diagram, we can see the steps involved in making a bacterial cell competent to take up recombinant DNA. In the first step, we can see that the bacteria is not readily taking up the recombinant DNA even though they are mixed together. Why? Because the bacteria has got a cell wall and a cell membrane which does not allow for the easy passage of recombinant DNA into the bacterial cell. In order to make the bacterial cell competent, we need to treat the bacterial cell with a particular concentration of divalent cation. right, such as calcium ion. This will make the bacterial cell wall porous to the recombinant DNA. And then we incubate the mixture on ice, followed by 42 degrees Celsius heat shock, and then back on ice. What this will do is, this will make the bacterial cell competent to take up the recombinant DNA into it, thereby transforming the bacteria. Right. So this is how you make the bacterial cells competent. And that is what we mean by a competent host. Apart from using cloning vectors, there are other ways to introduce alien DNA into host cells. So these are vectorless methods of introducing alien DNA into host cells. So the first one is microinjection. In this particular method, recombinant DNA or the desired gene is directly injected into the nucleus of an animal cell, thereby transforming the animal cell. And then we have biolistics or gene gun. So here is a gene gun. In this particular method, the plant cells, they are bombarded with high velocity microparticles of gold or tungsten coated with DNA. So if we want to introduce a desired gene into a plant cell, we can coat the DNA on these gold and tungsten microparticles and then shoot these microparticles at high velocity at the plant cells, thereby hoping the plant cells would get transformed. And then third, we have got disarmed pathogens. Right. Disarmed pathogens are also allowed to infect the cells to transfer the desired DNA into the host. So disarmed pathogen means the pathogens which are no longer virulent. They cannot cause disease, but what they can do is they can infect the cell and transfer the desired DNA or recombinant DNA into the host organism. Right. So these are some other ways to introduce alien DNA into host cells without the use of cloning vectors. With this, we have come to the end of this particular video. In the next video, we will discuss about processes of recombinant DNA technology. So you should read section 11.3. Okay, and then you can watch the next video. Thank you.